Thank you everyone for coming to today's Minds, Technology and Society seminar. I'm absolutely delighted to welcome Professor Virginia Dessaulx from the Cognitive Science Department at UC San Diego. Professor Dessaulx is a person whose work I have followed for many years, and I would say it kind of brings the best of interdisciplinary science, the kind that we've been talking at lunch earlier. So her work brings together cutting-edge neuroscientific techniques using EEG and computation with a lot of applications in things including brain-computer interfaces. Professor Dessaulx is the Associate Director of the Data Science Institute at UC San Diego as well. She got her undergraduate degree at Queen's University and her PhD at the University of Rochester, working with Dana Ballard, who was one of our distinguished cognitive scientists not that long ago. And then she did a postdoc with Jeff Hinton at the University of Toronto, and also a postdoc in neuroscience at UCSF. And so she's really brought the best of the worlds of computation and neuroscience together. And you're going to hear a lot more about it in her talk today, which is on recognizing, creating, and exploiting multiple views of EEG data. So please give a warm UC Merced welcome to Professor Dessaulx.
So much for that wonderful introduction. Okay, so I'm interested in the question of how humans and machines optimally extract information from their environment. I firmly believe that studying both human and machine learning is synergistic, and so I use insights from human brain physiology and behavior to guide development of machine learning algorithms, as well as using computational algorithms to extract information about human and animal brain processing. This talk is mostly going to be involving the latter because I think you guys are more interested in brain learning. So, that's what we're doing. So I've been interested in multi-view learning for a long time. This is actually my daughter many years ago. And I was interested in how it is that the brain is able to learn without a supervisory signal, and motivated by diagrams such as this one where you see all the visual areas, probably all seen this multiple times, multiple visual areas. But then up here we have these multi-sensory areas. And importantly, and kind of hidden in this diagram, is there's just as much feedback coming down as there is forward, and the question is, what does that do? And so I was interested in the question that this feedback might be very important for actually serving as a teaching signal for the visual system, and that likewise the visual system could form a teaching signal for the auditory system, and we made this very simplified version. And put some cows in there over here, but the idea being that two different sensory modalities could help to train each other and might provide useful teaching signals for each other. Okay, and this worked actually quite well to allow people, this was actually with ba, ba, da, ga, wa, way back when you had to use the little clapper to segment, to line up and synchronize video and auditory streams. But you could do almost as well as supervised learning just with this, looking at the co-occurrences and letting them label each other that way. Interestingly, the similar ideas also came up in supervised learning. This was joint work with Rich Caruana, where we looked at, let's say you have a problem, you have a whole bunch of input features, and you could take some of these input features, and instead of using them as inputs, you could make them be outputs. And if these are the output units you care about, this is the output labels you care about, you can actually do better sometimes when you've used these features here and you move them, instead of making them inputs, make them be outputs. So again, this idea of how do you optimally combine different kinds of information in order to get your system to learn properly. Okay, and so today I'm going to talk about how we are looking at finding multiple views in EEG data. And the first sub-project I'm going to talk about is, we can call it learning from noise in EEG-based motor imagery brain-computer interfaces. This is the PhD work of Mata Musavi, building on work from Adam Kerner, who was a neurosciences graduate student. Mata's an ECE student. We also had a visiting undergraduate, Chang Zhang, she was visiting undergrad, now she's a CMU PhD student. She might have even graduated, but she was a CMU PhD student at some point. And then the second part, we'll be talking about single trial prediction of memory responses at different stages of an episodic memory task. This was work started by Eun-Ho No, who was an ECE student. He graduated, and Kweda Lau is a current ECE student continuing the work, and is joint with Tim Curran, Britt Hertzman, Matt Mollison from UC Boulder. This was actually started as a Science of Learning Center project with the Temporal Dynamics of Learning Center, which was an NSF-funded Science of Learning Center. Okay, so brain-computer interfaces allow a user to bypass his muscles. Can I move this table forward a bit? Okay, allow a user to bypass his muscles and interact with the world directly through thinking, through brain activity. You can have invasive BCIs that involve recording under the skull. They've been very successful. You can see videos of monkeys using robotic arms. You can actually see videos now of humans controlling robotic arms. We work on non-invasive EEG, EEG-based control. And you probably don't need this slide, but EEG-based brain-computer interfaces detect voltages externally at the scalp. And so here's my little pyramidal neuron. If only one neuron receives input, you're not going to measure anything up here. But if many neurons that are similarly aligned receive near simultaneous input, then you can actually measure a voltage change up here at the scalp. Okay, and so because you're detecting voltage externally up here at the scalp, this obviously has advantages of avoiding opening up somebody's skull. You can do it on like undergraduates. But as the sensors are further removed from the neural activity, you need more careful task design and more sophisticated signal processing. So this makes it a much harder problem, makes it more interesting if you're into signal processing. And there's all kinds of different forms now that you can measure EEG in. This is the kind of signal you're looking at. You're looking to find with a motor imagery BCI. So and this is actual single trial data from actually a cognitive science grad student. Not everybody has data this nice. Not everybody's brain makes data this nice. But this is from channel C3, which is here. CZ in the middle, C4 over here. And you see this nice oscillation beforehand. That's called the mu rhythm. It's called the mu rhythm because it looks like the Greek letter mu. And during movement, prior to major movement, and during motor imagination, the mu rhythm decreases, the mu power decreases. It's called desynchronization. And this is a signal that you can use to control a brain-computer interface when you do the imagined movement. And then the idea is if you do right-hand imagery versus left-hand imagery, and you process it well, you should be able to tell whether they want to do right-hand imagery, whether they're doing right-hand imagery or left-hand imagery. Notice this person's doing right-hand imagery, and you actually see decrease on both sides. There is a little bit of timing difference. But it's actually much harder to get right-hand versus left-hand than just the fact that they're doing motor imagery. So it's harder to get that extra bit. But you can do that with spatial filtering to some degree, and so here's an example of a spatial filter. These little diagrams, the nose is sticking up, and there's ears here, so this is like the head. It looks like this extends beyond the head, but that's because the head is actually a sphere and not spherical, and this is a 2D flat thing, so the stuff on the edge is actually on the side of the head. And then what you can do is you can see the power in that 7 to 13 hertz bands over here. This might be the baseline in black, and then when they do motor imagery, you get this decrease in power in the mu band. And then higher frequencies, it's a little bit more than just the higher harmonics. This is not just the higher harmonics of that. Spatial filtering is commonly done with common spatial patterns. This is just a supervised dimensionality reduction algorithm where you're finding spatial filters that maximize the variance or power of one class relative to the power of the other class. And that's exactly those two classes. They differ not in mean, right? The mean is, these are bandpass signals, the mean is 0, but they differ in variance or power. And so that's why you need to basically transform into a classifier that distinguishes based on variance. Okay. All right, so first we have to do feature extraction, and we do that with the common spatial patterns. So here's some raw data. We will window it, so that's shown by this little light green rectangle. Then we can put it through the common spatial patterns. We take the power, and then we take the log of that because that's more normally distributed. And then if you do that, and here I'm just showing you the top 2 dimensions. Green might be, say, right-hand imagery. red might be left-hand imagery, and here you can see fairly representative data showing basically that there is some separation, but there's a lot of noise. Okay, so there's some information, but you're not going to do 100% a classifier. Is it a right-hand imagery or left-hand imagery? And this is usually within windows of about 500 milliseconds to a second long. So each one of those dots. Is everybody clear? Oh, feel free to interrupt and ask questions. And is everybody clear on this kind of outline? Because we're going to see a few of these. And we're also going to show, once we've done this, the classification boundary is very clear. So I'll also plot some where you have 1D representations, which is just the marginal distribution when you project onto here. Okay. So this is a plot from Krishna Shenoy's paper showing for the same subject, same electrode locations, just different blocks over time of the drift in the distributions of left-hand and right-hand imagery. And the line shows you the classification boundary that was learned in the first block and just how sometimes it's still fairly optimal and other times it's fairly suboptimal just because of the drift of these signals. Okay, so there's this non-stationarity due to a variety of things: sweating, movement, changes in attentional state, frustration. The losing of control itself can lead you to get frustrated and loss of, so basically loss of control can spiral out of control. And here's just here what I've done is taken that figure from Shenoy et al., just cut them out and put them on top so you can actually see them moving around over the blocks of time. Okay, so because we have noisy data, it's common to use multiple steps to hit a target, right? So if you're going to be a user using the system, you don't want a 65% accuracy to can make your wheelchair move. So, but you can chain together several steps, and then you can chain the accuracy to be arbitrarily, you can trade off time with accuracy to get an arbitrarily decent accuracy. And so that looks like this. The person might be trying to do right-hand imagery. You might get a step that's classified incorrectly, but then eventually it moves. And at some point, you will hit either the target or you hit the wrong side. And then you can look at the hit rate for accuracy. Okay, so we reasoned that this feedback itself from the BCI can be a large cause of non-stationarity. Right? So it's known that you train people in an offline manner, and then as soon as they start using the thing, well, all of a sudden the signals look different. And there's a cute example of this in an episode of The Big Bang Theory, which I hope you're somewhat familiar with. So in this episode, Sheldon's been dating Amy for a while, and he's very happy, and he feels like this is actually negatively impacting his work, right? So he's too happy, so he can't be creative enough. And so he's asked Amy as a neuroscientist to help him get into a better state so he can be more creative, right? So he feels like he has to be more anxious. So she is trying to make him more anxious, okay? And so this is it.
I don't like it, but that's the point of the experiment. I need to irritate you to find your optimal anxiety zone.
Perhaps this is a waste of time.
Sheldon, you're a remarkable scientist. Just be patient. I'm sure you will find the breakthrough you've been looking for.
Thank you.
And if you only do solid research instead of making a groundbreaking discovery, what does it matter?
Only do solid research? I come to you for help, and you insult me? I thought the least that you
Look,
would
your
do
anxiety levels are right in the zone.
Really? Oh, that's fantastic. Now, wait, they're dropping. Why are they dropping?
Because you're happy they're elevated.
Oh, that is infuriating. Yeah, terrific. Oh, no, they're going back down.
So this is a humorous example, but there's actually some truth in there too, right? When you're recording EEG, you're recording the whole brain activity. And so any brain activity that's going on there could be detected in your EEG and therefore can mess up your signal. So we figured that by explicitly modeling these effects, or at least some aspect of them, we might be able to improve the motor imagery classification. So we set up a system where we had 10 subjects, each with 200 trials and 10 blocks of 20 trials. And we made it, we call it fake online. So each block had a predetermined success rate, which varied from 60% to 90%. The reason for this is because we wanted to, normally there's this forward connection, like how good you do at motor imagery affects what you see, and we wanted to break that link because we wanted to control what you see and keep it separate from what you're generating, because we actually want to see how what you see affects your brain activity. So we can't have the cycle go both ways. We also wanted to we wanted to control the number of times it jumps towards the target, or good trials, versus the number of trials it jumps the other way, or bad trials. So each block had a predetermined success rate, but we set it up like it's a real-time BCI, and we give them decent success rates so they feel like they're controlling it, and the subjects thought they were controlling the BCI. Okay, so as we do this, then we can train a motor imagery classifier. So we can train the standard classifier to do right-hand imagery versus left-hand imagery. And these are the results for one particular subject for different frequency bands. So this is that the mu bands are here, and you'll see that these are better. You'll see again they're not 100%. This is for the 500 millisecond window. You're above chance. Chance is 50%, but then there's this error bar on chance, and that's the green line that depends on how many trials you have. And so this is a fairly good subject for right-left versus right-left motor imagery. And this is how well you do within each individual frequency band, given by here in hertz. If you combine this, like literally just a very simple averaging, you get the blue line. So this is how well you can do if you combine all of those classifiers. But we can also, at the same time, train a classifier to distinguish, did the cursor move in the good direction, i.e. the direction they wanted, or the bad direction, the other one. Okay? So it's different than right hand versus left hand. During a trial, they'll always be doing right hand, or they'll always be doing left hand based on whether it's they want to hit the right target or the left. But the good or bad is going to vary based on how the cursor moves, right? So if the cursor moved in this direction, we call that a good trial. If they want to go that way, it's a good trial. If they want to go the other way, that's a bad trial. So we train a classifier to distinguish good movements versus bad movements. If we do that for this particular subject, we get this dashed line here. So you'll notice that it is also above chance, and it's also good in the good frequency bands for motor imagery. So these signals are in the same frequency band. They can mess each other up if you ignore them. But if we use them, they can be helpful. And we can use them because the computer, actually made the cursor jump. So it knows what direction it went. So if it gets a good signal, it knows that you like that direction. If you get a bad, it means you don't like that direction. We can use that just as much as we can use the right, how well we detect the right versus left. So if we do that within each frequency band, we get the magenta curve, you see, which is generally better than the black. And overall, when we combine the magenta overall frequency bands, we get this new blue line, which is a little bit better. This is one subject, and this is our 10 subjects. So you see there's a wide variety in individual abilities. This subject is amazing at right versus left motor imagery. And he's still pretty good at good versus bad, but it doesn't help that much because he's already pretty good up there. There's other subjects who are pretty bad at right versus left motor imagery, like this one, but actually, somewhat okay at good versus bad, and the magenta can bring you up a little bit. People that are pretty good at each. Here's somebody who's not very good at right versus left, but much better at good versus bad, and the magenta actually tracks the better results of good versus bad, and here's another one like that down here. So large variety of differences, but the good-bad seems to either improve or not really hurt. And here is that, so this is the overall classification rate across all the frequency bands, and actually even including the standard GB ERP, so basically just the error potential. The red curve is just using right versus left, the red bar, and the black bar is using the right-left plus good-bad. And you'll see that some of the subjects are really not usable with just right-left, but everybody is pretty good. This would be the worst when you combine right-left and good-bad. So we've taken a signal that if you ignore what it's signaling, it actually adds noise and it messes up your right-left classification, but by paying attention to what it actually can signal and using it, we can actually do better. And so that's what I mean by using noise in the EEG signal. It's not really noise, although we call everything we don't want to measure, we call noise. But if you actually look at what it's recording, sometimes it can be useful. Okay, so yeah, response to cursor movement adds noise to the motor imagery signal. However, this noise is informative. And by modeling it instead of ignoring it, we were able to improve performance an average of 11% for single, in this case we're using 1-second window classifications. And the theoretical analyses also show that this good-bad signal is more robust in the face of non-optimal classification boundaries. So this is a subtle point that hopefully I can get across in a few minutes. Like I have a whole talk on this alone, but so you can model what's going, or the whole process with this Markov chain here. Okay? So if the person's doing left-hand imagery, then they're generating data from this Gaussian. So these are these one-dimensional distributions I said. If the person's doing right-hand imagery, they're generating from this. We have some classification boundary, and with probability A, we will move the cursor to the left, and with probability B, we'll move the cursor to the right. Okay? Now this is great if the classification boundary is in the optimal position as shown here. However, if the classification boundary is offset, which is going to be because you've seen that the data is moving around, so there is no one optimal boundary, then there's nothing you can do. A gets smaller, B gets bigger, so I kind of made these letters bigger here, and you're just going to, on right trials, it's going to be easier, on left trials, it's going to be really hard to get the left trials. Now contrast that to good-bad control. So here it's called continue. Continue, think of as good, and change as bad, right? So if it's good, you want it to continue in that direction. If it's bad, you want it to change direction. And these now are representing continue and change. So now if you have a left trial, it depends on the direction of the cursor movement, whether you generate a good or a bad signal, a continue or a change direction signal, right? So here the Markov chain now has states that not only depend on position, but also depend on cursor direction of movement. And likewise, the signal that the user is generating, if it's a left trial and the cursor is moving to the left, then they want to generate from the continue distribution, either A or B. On the other hand, if it's a left trial and the cursor is moving to the right, then you want to generate a change, and you're going to be generating D or C. Okay. Now look at what happens when we change the location of the boundary. You'll see that A got smaller, but C also got smaller, and D got bigger, and B got bigger. And notice B and D are pushing in different directions, and A and C are pushing in different directions. So there's a little bit of this trade-off. It's not as disastrous as A getting bigger and pushing one way, A getting smaller, B getting bigger. There's much more of this trade-off, and you can show that the information transfer rate at the optimal boundary, which would be here, they're identical, but this is the good bad, or this DS stands for dissatisfaction, satisfaction, good bad. So good bad control has a much higher information transfer rate as you move away from this optimal classification boundary than the standard right-left control does. So the good-bad signal, A, you're getting extra information from good-bad, but then it's also going to be much more robust to not having the optimal boundary. Okay, so we are continuing work in this area. We're working on improving the classification using Riemannian classifiers of the spatial and temporal covariance matrices. We're actually dealing with the proofs right now, so it's in press. So replacing the standard CSP method, the common spatial patterns method, with this new method improves classification of the GB signals for each subject, an average of about 8%. So we hope to put that into the new system and it'll do even better overall. We're developing convolutional neural networks of common spatial patterns and combining that with and also doing window means classifiers. This is not a huge win, but putting the common spatial patterns into one network has an advantage of preventing overfitting. So the common spatial pattern algorithm, it's a supervised algorithm, and there's no easy way to prevent overfitting at that stage. But if you put it into the whole deep learning framework, then you can use the standard techniques of deep learning to prevent overfitting overall. So there's a little bit of win there. And then we're also working on looking at unsupervised transfer learning algorithms. So all of these were trained on a per subject basis, but it's interesting to be able to move classifiers from one subject to another. And now, excitingly, we're testing online versions, so real online, that directly and indirectly use the good-bad signals. And we are finding that good-bad control is working. When we allow the good-bad to help control, they do a lot better. This we have not started yet, but I'm very interested in using the known relationship of good bad signals for self-supervised learning. Okay, so we're gonna switch gears to the next project, so if you had any dying questions you could ask now, or we can wait to the end. Okay. So, okay, so our next project we're gonna talk about predicting memory performance. And here our big picture goal was, can we help people study? Can we present stimuli at optimal times for learning? And after presenting, can we tell whether they will be remembering? Okay, so it started as kind of an engineering interesting question. It was actually a question that came up in a Temporal Dynamics of Learning Center meeting. It's like, what can you do with the TDLC that you couldn't do by yourself? And so there were lots of people studying memory in EEG, so I thought, well, let's try this. Okay, so the idea being, so the EEG thing comes along and you want to predict the optimal time for encoding. That was the motivation. Okay, so we started, oops, I skipped one. So our first step was, can we find EEG signatures that indicate a person is in a good brain state for remembering? That's the first part of what we're going to talk about. And the second one, can we find EEG signatures that indicate encoding was well performed? We'll look at that also. And as our eventual goals for practical use, we search for EEG signatures that can be found in real time on single trials. We have not run this in real time in an online system, but we're doing single trial classification. Okay, so the data were collected by Tim Curran and Britt Hertzman many years prior, so obviously not online, in an episodic recognition memory experiment. And so they had a study phase and a test phase, a recognition phase. And they were presented with pictures. These are pictures of cars and pictures of birds. And during the test phase, they have to give one of these 5 labels to the tested images. Recollect, definitely familiar, maybe unfamiliar, and definitely unfamiliar. Importantly, recollect responses the subjects were told are to be given when the subjects had a conscious recollection of learning the item in the study phase. Okay, so recollection is like, I see Spivey, I can remember him from grad school, it's like all kinds of good memories, etc. Familiarity would be, I like see him like, he looks familiar. Where have I seen him? That's familiarity. But recollection is you have this conscious recollection of learning the item. Okay. So we trained a classifier, and to start with, we ignored the familiar. So we trained recollect versus unfamiliar. You know, we would have ignored maybe unfamiliar, except we didn't have enough unfamiliar trials. So we're going to train recollect versus the 2 unfamiliars. Okay, that's going to be our classification problem. Okay. And there's been lots of prior work on this. So the Gurdarian et al. Has looked, this is a MEG study looking at changes in theta power predictive of future memory performance. This is an ECOG study showing changes over a large range of frequencies, again, predictive of memory performance. The difference is that these were done not on single trial basis, but like ERP studies, averaging over many trials. This is the difference due to memory, or an ERP, seen in the during period. And this is another during period result showing changes in alpha power. So based on these, we decided what we were going to look for in single trial. In the this is data from the original experiment, so the subjects are all right-handed males. They're all male because they were interested in issues of car experts and bird experts, and it's really hard to find a female car expert. So all other subjects were males. So these are detailed visual memory, 8 blocks, pictures of cars and odd blocks, birds and even blocks. And the study phase is 40 targets to be memorized per block, and the recognition phase has the targets discriminated from 40 new distractors in random order. Okay, so we generated 3 separate classifiers, and some of them had subparts. So we looked at power prior to presentation in these frequency bands. We looked at the single trial instantiation of the ERP after presentation. And we looked at power in the alpha band in an early period, and then after the reviewers told us, we also looked at a later period as well. So you see that. And then we combine all of this to predict whether the subject will remember or forget the item. Okay, so this is just showing subfields. So we trained the individual frequency bands. We did a nested cross-validation to decide whether to include those frequency bands in the final classifier. This doesn't matter too much though. And then we trained different classifiers for the temporal signal and then the alpha in 2 different time ranges. The classifiers are all very simple though, just linear, I mean, the CSPs and then the standard simple, because the signals are so noisy, you don't want to do anything too fancy. Again, we see large differences in human response. So this is not how well the humans remembered. This is how well we are able to classify whether they will remember. Particularly if the human was excellent and remembered everything, then we couldn't even do them because we wouldn't have any forgotten trials, right? So these are moderately performing humans. So if they were too good, we didn't have enough data on forgotten. If they were too bad, we threw them out because these are just undergrads, so they probably weren't paying any attention. So these are sort of like middle subjects for memory performance, but we have different amounts of ability to tell. Often this is actually fairly well correlated with how much data we have from them too. So the ones that we do better on, we tend to have more data. The ones we don't do so well, we tend to have less data. And the stars represent ones that are significantly above chance. Okay, so this from an engineering point of view is somewhat interesting, but we decided to go in and look more at what was going on. And one thing we can look at is we trained classifiers on the individual frequency bands. In this is all in the prior period, right? So prior, before they even see the item. And so this is the response for recollect in the test data versus maybe and definitely unfamiliar, shown in black with the circles and stars down here. You know, showing basically some separation. But now we can plot the familiars. So we can see what did they do with the familiars. And when we do that, we see that in the lower frequency bands, the familiars look like the recollect. And in the higher frequency bands, the familiars look like the unfamiliar. So this is interesting because it looks like in the low frequency bands, we're classifying based on familiarity. And in the higher frequency bands, we're classifying based on recollection. Okay. So that's kind of cool. We didn't tell it to do that, but that's just what it's picking out. We can also look at the 2 different time periods where we were looking at alpha. So early on in the 400 to 800 millisecond, definitely familiar looks somewhat like recollect. Later on, definitely familiar looks like the unfamiliar. So again, it looks like early on we might be classifying based on familiarity, later on recollection. This makes somewhat intuitive sense of first you have to recognize the item and then you can bring in all that context that's important for recollection. There are actually some consistencies across subjects. So these are plots showing what comes out as consistent across subjects in different frequency bands and time periods. So this is theta 200 to 600 milliseconds, alpha 400 to 800 milliseconds, alpha in the later period, and then low beta in the middle period. But alpha and beta feel like this pattern should be abolished from this to that, looking very much like that in between. So these are plots of different time periods, but also different frequencies, but there's a lot of overlap there. So the data were already pre-collected when we decided to classify them. And in many ways, doing an offline experiment is a lot easier than doing an online experiment. But in one way, it's harder, right? So if we were doing an online experiment, what we would be doing is we'd be monitoring the EEG, and then we'd be presenting items when we thought they were in an optimal brain state for remembering, and we might compare that to when we thought they were in the least optimal brain state. But here we just they're already pre-computed, so it has nothing to do with their brain state. So we can simulate that, and so what we can do is we can take the score of our classifier before we threshold it, and take the top 10% and say, these are what we think are the best brain states. And we can take the bottom 10% and say, these are when we think it's the worst 10% brain states. And we compare how well they do behaviorally in remembering the items from the best brain states as measured by our classifier, versus the worst brain states as measured by our classifier. So, we're simulating this, triggering these for good brain states and bad brain states. If we do that, we get these results. So this is predicted good states. They are remembering a little over 60%, and in the predicted bad states, they're remembering a little less than 40%. The 10% doesn't matter that much, like if you vary the ratio, you just get this, like if this is all of the data, then you sort of just spread out like this as you include more or less of it as you go this way. So, it's not like we cherry pick the 10% to get that. This is somewhat similar to, this is a paper by Yu et al., which is a real-time fMRI study where they were looking at memory for places by monitoring bold response in parietal place area. And so you see the EEG is doing at least as well as the fMRI. I mean, this is a real online study, so they're not directly comparable, but it looks like there's potential there. So results from our predicting memory study show pre-stimulus brain state represented by high beta, low gamma power may play a role in improving the context plus item encoding. And this is consistent with some other results that finding beta and gamma power prior to stimulus presentation reflected states to enhance attention. Pre-stimulus brain state represented by lower frequency seems to reflect learning of the item. And the during stimulus alpha power originally coded for familiar versus unfamiliar, and later seemed to separate recollect from familiar, implying that encoding of context may occur after encoding an item, which kind of you couldn't really expect anything else, but interesting that it shows that. Okay. We can also look at responses during the test phase. So we can take data and look at responses during the test phase and see whether we can again see evidence of whether or not they'll say that they remembered an item. And there again, there is prior work looking at ERP old new effects. So there's the parietal old new effect, which is this positive going ERP, typically observed between 500 and 800 milliseconds on left lateral eyes. Shown here is the difference between the yellow, yellow and the black over here. And the frontal old new effect, which seems to be more related to familiarity. And then there's also this late posterior negativity, which is later than the two above and more negative for correct old, whether the new responses, irrespective of the information information accuracy. Okay, so that's shown here. So we're going to look for these three signals, so that affects the time range we look at and the electrodes we're going to be looking over. Okay, and we're going to use data also from Tim Kern's lab, but this was with Matt Mollison. We couldn't use data from the first experiment because they were responding with their hands and they were using a different hand for remembered than forgotten, and so we weren't positive that we could remove the motor aspect of the task from any memory aspect we wanted to say. Another big problem with using EEG, right? You're recording everything. So we just went with this one where the responses are much delayed, and so this is a different task, a different memory task. This one, they were told to they weren't using this recollect from familiar, they were actually told to remember specific, what's called source memory. I don't like that term, but that's the way the field is. So they have to remember, this is the spatial location version, they have to remember which side of the screen the item was seen on. So they remember the item and they remember the side of the screen, so right or left. Then at test time, they have to, if they remember the item, they have to say whether it was on the right or the left. And if they get it correct, and they get the right or the left part correct, it's called a correct source judgment, SC. If they get the item correct, but they get the side wrong, so this was really on the right, they said left, we call this incorrect source judgment, or SI. Could be a few of these letters. And then if the dog, and they say it's new, so if they think they haven't seen it, then they say new, and this will be a miss. You can also have correct rejections and false alarms. Okay. There's also another version where the item, where the source information. Okay. Oh, so this is the ordering. So you see the item, then you have left plus right or new, and then after you say this left, right, or new, you have to say, if you pick left or right, that means you think you saw it before. Here you have to say whether you believe that you recollect the source, or you recollect something else about it. So you know you recollect it, but you don't actually remember what side it was on, but you definitely remember seeing it, or it's just familiar. If you thought it was new, you have to say whether you're sure or maybe. So sure, new, or maybe new. And then this is the side version, and then there's a colored outline version where you have to remember the color of the box that's on the outside from a choice of 2. Okay. Yeah. Let me raise his eyebrows, and this is actually harder, and you'll see there's some interesting aspect to that. But here, again, you first say red or blue or new, and then you say recollect color, recollect other, or familiar. We're going to just change these to RS, RO. So recollect source, recollect other, or familiar, or sure or maybe new. Okay? So that gives us 13 different trial types. These will all get used in a second. So we have source correct, so this is they got the source right. And then here they knew that they got it right. They don't think they got it right, and they really are kind of just guessing. Source incorrect, they thought they got it right. They knew, or they think they didn't get it right, or they think they don't know it, and they're just guessing. False alarm, same thing. So here they think they see it and exactly. Correct rejects, maybe new, sure new, and misses, maybe new, sure new. Okay, so classification is performed using LDA, linear discriminant analysis, on spatially averaged. So these are really simple classifiers. We're just averaging over time and averaging over electrodes. Within the 6 defined clusters, time averaged, yeah. Okay. And we started by classifying SC versus CR. So source correct versus correct reject. Like again, the strongest classes. We're also going to look at, you'll see SC versus SI and SI versus CR. Classification results are slightly better than you get from training on the training data. So when you're using this data at test time, you can do a little bit better than when you use the data from training time. Again, you're not perfect. Average AUC results across all subjects varied from 0.59. They also had some experiments where they actually brought them back a second day, and so we're a little lower on those. Again, the electrodes are probably in slightly different places. And we're able to train yeah, you'll see. So we're actually able to train a universal classifier, which you'll see. And this allows us also to train the 13 behavioral classifiers against each other, which you're going to see in a second. Yeah, so some of this slide is a little out of order for because I added extra stuff. So these are the results. Again, for different subjects, again, fairly large variability. The bolded ones are significantly above chance. This is SC versus CR. SI versus CR, SC versus SI. This is for the spatial location one. One thing to note is SCCR obviously is the best performing. Like some of them are up to 74, 75%. And there's more bolded in this column. There's also more bolded in this column than this one. This is SC versus SI, whether they get that source information correct for the side one. And not that much for SI-CR. For the color one, it switches. For color, the SI versus CR is more distinguishable than SC versus SI. And you'll see in a second that's interesting. The color one is a little bit harder. We can do the same thing we did with the other data set. So here the trained classes are circled. Again, it's test data but from the same class. So this is trained on SC versus CR. This is where it puts SI in between. That's kind of nice. The misses look like correct rejects. It's also kind of nice. And the false alarms look a lot like the SIs. Basically the familiar stuff. If we just train an SC versus SI classifier, this is averaged over the side and the color versions. Everything looks like SI, all the other ones, which makes sense because there is no source information. It'd be very strange if it actually looked like an SC. If we train SI versus CR, you'll see, you might think, well, from the other results, you might say, well, you trained it on these two classes, everybody else is going to fall in between, but that's not the case here. You see SCs up here, which is nice. So SC looks like SI, or maybe even stronger, further away from correct reject. Miss, again, looks like correct reject. The false alarm, again, looks like source incorrect. That's just the three of them together. You can look at the patterns. These are those areas on the brain up here, LAS. Again, the nose is here, the ears are here. just showing you the areas in which direction they go for SC versus CR, SI versus CR, SC versus SI. This is average over both of those color inside. Interestingly, the SI-CR seems to be more on the early side. Time is here. SC versus SI is later. Again, first you get the familiarity, the item info, and then you get the source information. This is if we split it out by, experiment 1 was the spatial one, experiment 2 is the color, and experiment 3 actually had the same subjects doing color and space, and spread over 2 days. Not color one day and space one day, but color and space one day, and that's not enough trials, so then they bring them back and they do the other half of color and space. So 3 is harder, but you see a little bit of pattern here. SC versus SI is much stronger for the location, and SI versus CR tends to be a little stronger on the colors. So there's definitely a little bit of difference. It seems that from here you can say that, and this again should make some sense to you, the side that the item was on, that comes with the item more for free, almost for free, because there's not as much signal here, but basically if you get the item, you tend to get the source correct. But the color one, that was much harder to remember, like the color. And that makes sense, it's not as natural, right? It's not as a natural thing to, who cares what the color of the outline is. But the side that the item was on, that's often used as a memory aid, actually, is to remember where things were. Okay, now, again, like the beginning work, we trained everything on individual subjects. However, this plot is, again, what's consistent over subjects. So we've decided that for these simple classifiers, we would look and see if we could actually do a leave one subject out classifier. So instead of leaving one trial out classifier the standard way, we'll leave a subject out. And here you see leave one subject out compared to leave one trial. So this is leave one subject out, leave one trial out. There's a trade-off here. Leave one trial out means you have less. As soon as you go to leave one subject out, you get tons of data, right? Because you can combine all those subjects. We have a lot of subjects here. On the other hand, it's data from like people that are quite like you. So there's that trade-off between better fit versus more data. And you'll see that the trade-off is fairly even. So we do about as well with leave one trial out as we do with leave one subject out, except for the SC versus CR, we do better with leave one trial out because it tends to be more data here. But I mean, generally, we do about the same with leave one subject out. We can even go further than this in that we can, so here we've got an SC versus CR, SI versus CR, and SC versus SI. We can look at how well the SC-CR classifier does, train leave one subject on all of the other ones. So that's shown here. We train the classifier on SC versus CR. We can test data from a different subject or the same subject on SC versus CR. We can train on SC versus SI and see how well does that classifier classify on those two. It might classify well, it might classify badly. But if it classifies well, it says that, well, those are pretty much aligned. And SI might be just kind of in between SC and CR. And that appears to be what we see, largely the first degree here. So here we have classifiers trained on SC versus CR, SI versus CR, SC versus SI, versus behaviors. So this is the test data, and this is the classifier. It's a little confusing. But so this is SCCR, trained on SCCR, tested on SCCR. Again, not the same data, but done cross-validation. And that's where you get the best results. But here, this is using the SCCR classifier to do an SICR classification. We do pretty much as well as we do training SICR. And likewise for SCSI, we do pretty much as well. The darkness of that orangey color is supposed to tell you how well it does. So basically, training one SCCR classifier with all the subjects in does pretty well on all of the classifications, meaning that SI is kind of in between SC and CR, especially when you go with all the differences across subjects and everything thrown into here. Okay. Now, I said we had 13 different trial types. So we can actually, now that we've thrown in, on individual subjects, we don't have enough of any one particular trial type to do much with all of these different divisions. But now that we've combined data across subjects, we can actually start to look at all of these different trial types. So what we decided to do was to train every one of these classes versus every other one of these classes. Okay, so we train one class on SCRS versus SCRO, we train another classifier on FARS versus SCF, you know, each combination of these we can run a classifier. That will then give us a bunch of new dimensions, which we can then take in the individual trial, project it onto that classifier. So just like back here, you can take it, project it on the classifier. Before you threshold, you can basically get a score, which shows how far along are you on that one-dimensional projection. Right, so each of these classifiers of the 13 different trial types against each other now gives us a new projection. So essentially we take each trial and we blow it up into this high D space where each one of those dimensions is one of these classes versus another class. Then we can run PCA on that and see what do we get. Okay. So that's shown here. And these are the means now of the classes in the PCA of this space. So the top 3 dimensions. And I've color-coded them here. So this is SCRS, FARS, SI. So here they're colored based on confidence, right? So sure, new, and recollect source. Those are like, sure new is I'm sure it's new, recollect source is I'm sure I remember it. I remember it, I know I remember it. So those are blue. Red is kind of what I call the wishy-washy, right? So maybe news and familiar. It's kind of like, yeah, maybe. And then magenta is kind of in between. That's that RO, recollect. I recollected, but I don't remember the actual color or source. And so here you can see, okay, they're largely separating out red from blue. I think I can like make this thing spin and you can actually see a little bit better the 3D. The magentas are sometimes a little mixed in, but basically there's a separation between red and blue. Again, this is like just running PCA on this space. Okay, now we can take the same data and relabel. So now we're going to label it based on how much memory you have of the item, right? So here we're changing, so now on one end we have correct, reject, sure, new. So you're sure you haven't seen it. That's red. And then we have maybe sure, new, that's orange. Then yellow is this false alarm color, and then green is SI, and then blue is SD. So blue is you got it right, you got it right, you got the item and the source right. Green is you got the item but not the source right. And then, you said you knew that you remembered them. And then the red and orange are either misses or correct rejects. So you said they were new. So red and orange, you believe you have not seen the item before. Blue, green, yellow is you believe you have seen the item before. Okay. So this is a different way of dividing it. It's like, do I think it's new or do I not think it's new? And you'll see that that's a clear division here. And then we can look at the different experiments. So this is experiment 1, this is experiment 2. Again, this is the red, magenta, blue. So confident is in blue, not confident is in red. And then if we color it the other way, think you've seen it before versus think it's new in red, orange. And so here's experiment 1, this experiment 2, experiment 3, spatial, and experiment 3, color. Coloring it this way again, so basically each one of these, and these spaces are done totally separately, but each one of them has this division that shows, okay, this is the signal that says whether, that basically predicts whether I'm going to say I've seen it before or not. And at the same time, there's another division that's almost perpendicular that says whether or not I feel it's confident or not. And this is kind of interesting because some people say that recollect is just extra confident familiarity. But here we've also got confidence that it's new. And the confidence that it's new is not that far away from the confidence. So this is confident that it's old and this is confident that it's new, and yet there's this division of confidence that's separate from actual memory strength. So anyway, we thought that was pretty interesting, and it comes out in all of the experiments pretty much independently. Okay, so we're interested in further exploring the relationship between confidence and memory strength, and we're kind of new to this field, so we have a lot to learn. And we're relating signals in the test phase to those in the training phase, so this is Schwantan Feng is an undergraduate who's interested in looking at that with CLIA. So this new data set that we have, we can actually go and look at the study phase as well. So we're going to look at how to relate those. We're also examining differences with aging, which is a collaborative effort we started with Marta Kudas's lab. There are very big differences in ERP between older and younger subjects. Okay, and so overall, there are many signals multiplexed in natural data. Machine learning approaches can be used to pull some of this out. EEG contains useful information about the user's response to the feedback in many frequency bands. Trained classification algorithms can reveal interesting structure in the memory signals. And let's see, what time is that? I just had this slide from another talk at the rail. We'll leave it in. We're also interested in improving machine learning through inspiration from cortex, so incorporating lateral current connections and deep convolutional neural nets. So these are just images that if you just run a feed-forward standard deep network, you pull up all the texture edges. But if you now have recurrent horizontal connections, you can actually just evolve it and you can learn to get more of edges that are more relevant for determining objects. So we're interested in looking at that. And these are the acknowledgements, different people involved in these particular projects, different funders or supporters, and just overall people in my lab and overall support shown here. Thank you.
We have plenty of time for questions, beginning with student questions first.
It was all totally clear. Or totally opaque and you don't know where to start.
We'll open the floor up to anyone who wants to have a question.
You hinted at the distinction between familiarity and confident recollection, and then some memory researchers want to say it's maybe one mechanism and familiarity is just not quite confident recollection.
Right.
And there's other memory researchers who want to suggest that these are two independent mechanisms
Right.
that might have orthogonal dimensions when you PCA them. I want to get you to connect that to the earlier findings of the time difference that when, so low frequency and early on, familiar trials show EEG data that pattern with recollection trials. And then higher frequency and about a second later, all of a sudden those familiar trials
So it's a higher frequency in the pre-period,
Yeah.
and then about a second later in the during period, as
Yeah.
we call it, yeah.
And do you feel like this is evidence for sort of independent, or you wouldn't say independent, but semi-separate mechanisms for familiarity doing one kind of thing and genuine recollection doing
Yeah.
a different kind of thing?
We have not wanted to wade into that debate. I think that there's a little bit of both. And even the PCA stuff doesn't clearly, it's kind of orthogonal to that argument even, because it doesn't clearly show that familiarity is extra strong recollection. We have a little bit of evidence that pulls you in both directions for that. And then the PCA stuff is kind of a whole new other thing saying, well, actually there's this thing people haven't even talked about, which is confidence in your answer that's separate from confidence in the memory strength. So I'm not going to. I also don't know. I mean, I'm very new to memory, so I'm not going to dive into that debate and give an answer. I will say though that at least once this particular one is kind of saying that. Yeah, and then the whole way you do the task is different too, right? So the source incorrect is a little bit different than making the familiar judgment. All I'll say is that this particular result kind of implies that the SI class is in between the SC and the CR class, but that's when you average over all subjects. So within a subject, there might be a little bit off. By the time you look at all the variations across subjects, then the SI looks like it's in between the SC and the CR. But I don't have a strong view as to which of those other views is, and I think that there's this orthogonal, this other question, which is that there does seem to be something in confidence that's totally separate and hasn't even been discussed, about that there's actually confidence in that it's new and confidence that it's old, and there's something in there that's actually very related. Yeah.
So in the earlier experiment with the dot moving back and forth to the left or the right, part of the punchline there is that this good-bad measure is influencing the signal that you're getting and can be used to make this discrimination.
Yeah.
In this latter memory work, people are actually reporting their confidence, right?
Yes.
So this raises the question of whether the preparation of the report is generating part of the signal that's allowing you to make these discriminations. And unfortunately, I can't see how you could, unlike the good, bad, you can't kind of predict what their confidence report would be without asking them to think about it, right?
So for this one, they first respond whether it's new or whether it was on the right or the left, and then after that they respond with more of this confidence measure. And
they know that's coming.
they know that's coming, so yes, they could be starting to plan it, and it might be part of it. But yeah, it's like how to move it.
It sounds hard.
But we're still finding some interesting separations here that I think are worth looking at.
It just seems like it would be interesting to try to have like an implicit measure of confidence or something like that and compare the data in that domain with the explicit reports of confidence just to see if everything is the
that
same.
would be. Tim's lab has a whole bunch of other data sets that we could go back and look and see if there's other different aspects that might. If we can look at ones that have slightly different timing, we might be able to separate out things that way.
Right.
Yeah. Yes.
I'm curious to hear a little bit more about the work you're doing with Marta Casas on aging.
Oh, aging. So she has data from many years back. In fact, some of these subjects have since died, and some of these subjects are actually still alive, and you can kind of actually see. This is being video, so I won't say it. But yeah, so she has subjects, and at the time, even many years back, they were older subjects, and then there were younger who were largely college age, but then there were some older subjects. And she had them come in, and they were doing a task. They were remembering endings of sentences, and some of them were sensible endings, and some of them were non-sensible endings, and some of them had high close probability, and some of them had low close probability. And then if you just look at the memory performance, the older subjects did very poorly on the strange endings relative to the younger ones. And even if she did a pretest, like a standard memory, I forget, maybe there's a standard, maybe there's standard memory tests. So she did like a standard working memory level test, and they were, the older people were a little bit worse than the younger people, but not hugely off. And you could even control and take ones that were actually in the same range and compare. And yet within this range, those older subjects did a lot worse on the strange endings than the younger subjects did, like way, way worse, like very distributed. I don't think there was even any overlap. And then at the same time, the ERPs in the older subjects are much more smoothed out and blurred, like kind of like low pass filtered. And they had collected these data and they hadn't really done much with them, and we were in part of a project where we were supposed to look at aging memory related. It was an aging related big project. We could talk about later what happened to it. But as a result, we were going to start by taking data that was already collected and looking and analyze it. And Yeah, and so we could take the data and we can actually classify age. Marta being Marta goes, why do you need to do that? You just ask them how old they are, which is true. But it's interesting that you can actually see from the EEG, you can predict age quite well. And then we're interested, well, actually, can you predict memory performance, or can you predict like, brain age or whatever, relative brain age? And so we're kind of going down that path. We haven't done that much on it recently, but there's a lot of interesting information. You can definitely get a very nice strong correlation for the classifier being able to pick out brain age. And then these different between the congruent and the incongruent endings. It was a different classifier for the older and the younger subjects. It's just very different. See, the older subjects seem to, they even did a little bit better on the congruent endings. It's kind of like they were like basically they've had many years of experience with congruent endings, so they're very good at that. The incongruent ones, they just like ignore them. They just don't remember it.
Yeah. Oh. Sorry, Zoe. Well, part of the reason why I was asking about it is because Randy McIntosh at the Rotman in Toronto has done some work showing that as we age, there's kind of this U-shape, inverted U-shape function. When we're young, we have a lot of brain signal variability. When we're old, we also have a lot of brain signal variability. And so I was kind of wondering, with respect to your classification analyses, if it's somehow being affected or picking up these differences, age-related differences in brain signal variability in terms of like the EEG.
We were not so much looking at, it was more, so we're just learning like basically mean classifiers. These are old, these are young. We weren't even doing a regression, although in the end we can plot out the regression. So we were not, it's possible that we could look at that, but we weren't even looking at that. We were just looking at the differences between the older population and the younger population. But there were quite large differences. It's very interesting. And these are the older subjects at the time, these are volunteers who say, they're largely highly educated UCSD ex-professor types who are part of, there's a couple of groups that have very involved older faculty, and they go to talks and stuff, and there's a large amount of taking of subjects from that pool. So they're highly educated. I guess the younger subjects are also college students, so they're all highly educated. But they're pretty highly educated on both ends. So, and we're still seeing these differences between the older adults and the younger. And they're active because they came in to do these for a minute.
I don't know if you know, but when the elderly patients did poorly on the weird sentences, was it because they were confabulating to make them normal, or just not knowing an answer at all?
So they were told to fill in, they were supposed to fill in the word that they had seen, and if they didn't remember the word, then they were to just guess. No, they were supposed to guess. So,
so they
I think that they were filling in
sensible configurations.
Sensible configurations. And I don't remember that for sure, but yeah. But they just were really not able to remember those strange things. It's actually very hard. I mean, I've looked at it. When I give that talk, it was like you show them that they're hard to remember. They don't make much sense. But I think it's not a memory deficit on their part. I think they've actually, because they've stored so much other stuff, it's just inconsistent with all the rest of the stuff, and it doesn't fit as well. Yes?
So in sort of translating this work into building brain-computer interfaces that will turn things on, drive cars from EEG data, it strikes me like one of the missing pieces is being able to gauge the subject's level of confidence in their own performance, right, to some extent. I want to turn the light on. There's a lot of variability in that signal.
Yes.
So your classifiers are doing a good job in trying to get at what that signal is, if it knows how confident the person is. But I'm just wondering, like, is there a way of mining that sort of bottom up just from the signal itself, and then using that to drive these external devices?
Well, you can use like the error type signal, right? So if you could have a system, whatever system you have, and so it detects that it thinks you want to turn the lights on, and so it turns the lights on, and then if you get an error response, then it could detect that and turn that off. And so the error responses are pretty natural and don't take any work on your behalf, right? And so generally, whereas the motor imagery is a fair amount of work when you're first learning it, although it becomes more natural, and so people who are really good at it, they're just kind of doing it as not very, it's very exhausting when you're first learning it, but apparently it becomes much more natural. But the error signal is totally natural and free. You get it for free. And so you can use those as error signals. You know, think about the self-driving cars. When you're about to hit something, or when you think you're about to hit something, there's a full body response, right? That's just a pretty easy signal to measure, and so you could use that for the car as like to stop what it's doing, or probably don't just want to stop the car, but at least, you know, prepare the car, set the suspension better, be ready to brake, you know, get the brake pedal really sensitive, etc. So those kind of freebie signals, I think, are very useful, yeah. But you're never going to be 100% perfect. So you don't want to be, apparently there was some politician who thought that BCIs for shooting guns would be good because then nobody would know which soldier shot the gun. But that would be a bad idea. But you can use stuff, you can use it to make decisions that don't matter so much whether you get it right or not, or you know, on that particular trial. Like turning the lights on, it's not a big deal. You turn the light on. Yeah, they didn't want the light on, it's not that big a deal, especially if you can recognize the error and undo it after the fact.
Following up on that, given that it's kind of difficult to do the left-right motor imagery, what's the history behind using that task? Why not explore tasks that might produce a much larger overall brain difference?
So people have tried other tasks, and some, so mental rotation is used, internal counting. Gary Mackay actually says he was a subject in his dad's experiments, and he was getting really bored, so he was counting, and his dad came in and said, "Are you counting?" And he's like, "Wow." So there are, but they're usually not as informative as the motor imagery. So the motor imagery is used because people knew about this mu desynchronization, and they could see it, and it happens when you move, and then the idea is therefore you can do it. But it works really well for some people and not for others, and then there were some studies in Europe trying to figure out what it was about the subject that made it work well for these subjects and not those, and but they've never really been able to figure it out. And partly it's, you know, how your brain is folded, right? So you could be generating a great signal, but if the dipoles are not aligned appropriately, it's not going to be detected with EEG very well. And then, you know, it's also how well you're actually able to generate those signals. So, yeah, there was a whole study in Germany, I think, Germany or Austria, where they were trying to run a whole bunch of regular people through a bunch of different BCI paradigms and see what worked for what. But I don't think that they ever really decided this works for this and this works for this. And then there's people say, you know, if you're good at sports, then you can do good motor imagery, or if you're good at music, then you can do good motor imagery. We did have two identical twins who had fairly similar looking EEG responses, but.
Has any work been done like this in a virtual environment? Because I imagine that creating that, you get that good correlation of, like, you know what their brain can do, the EEG signal for what they want to do, and then you get them actually, you know, pressing a button or whatever they want to do to control the avatar. And so, like, is there any work?
So there's work showing that, yeah, the better, so you can actually get these desynchronizations also when you see somebody else. So there's supposed to be the human analog of mirror neuron activity. So you can also get this mu desynchronization when you observe someone else doing movements. And so one of my first graduate student who pulled us into brain-computer interfaces, he was interested in if we give better feedback, can we actually get a positive loop going. And so he was working on, instead of having a dot move to the right or the left, he actually saw this hand You see I think you see the hand, and the hand was doing movements. And so the idea being if you actually saw the hand moving, that would give you further desynchronization. So there's a little bit of work working on more in a more immersive, even more than that, system. So people are interested in that. Brain-computer interface environments where they put you in an immersive environment to give better signals. Yeah. We haven't done that recently, but.
I also imagine that something that might help training your subjects to the imagery task is to work on their attentional systems to some extent, right? Like,
Yes.
So telling them what to focus on in the task and.
Very much so, yeah. And my student Mata, who's leading this motor imagery, part of what she did actually was work on the motor imagery training, and we did some. So right now, usually you come in and you're told to do right-hand imagery versus left-hand imagery, and often you're just told like the classification answer. So you're sort of basically trained the difference between the right imagery and the left imagery, and you don't know. So when you're doing right-hand imagery, you don't know if you're not successful because both sides are desynchronizing, and so the difference isn't that well, or neither side is desynchronizing. And we reasoned that would actually be a huge deal, right? Because you might be trying too hard, in which case you're desynchronizing, well, right imagery would be desynchronizing over here, but you might also be desynchronizing over there, or you might not be trying hard enough, in which case neither is desynchronizing. So we tried showing them 2 bars, and then because we didn't want them to deal with the contralateral organization, we kind of showed the, and we were looking for decrease, but we told them we're looking for, you want the bar big on your side. And so we show the bars on both sides and say, you want this bar high, this bar low when you're doing the right, and you vice versa. And it looked, we didn't have a ton of subject, but it did look like they were doing better when we gave them that stimulus than just giving them the difference between the 2 bars, which makes sense. Right? We're giving them more information. So we believe, it's hard, right? Because you bring them in and you say, just do this imagery, and it either works or it doesn't work. That's not very helpful. So it's a little bit better to give them a little bit more information. You know, you want this bar high, you want this bar low. It gives them a little bit more for them to play with. And we even told them, if they're both high, then you need to do this, and if they're both low, so in one case, so if they're both high, that means you haven't desynced either. That means you need to try harder, and if they're both low, then just try more to lateralize it more. So we should present that at SFM, but we haven't published it. But yeah, it seems like working on training the subjects would be like a big deal.
All right, if there are no further questions, we thank Professor Soft.